Thank you all for coming. It's delightful to have a nice big filled room and be able to be in person and to be able to introduce Prashanth Rangan. So Prash and I first met in 2013, I believe, at a Dev Bioborn conference.
Yeah, because the bigger key opens that room.
Like a year or 2 years. And we gravitated towards each other, or I'm going to speak for both of us, which, you know, whatever. So I can flatter myself, I flatter him, because we both are sort of driven by sort of similar questions in terms of how do cells make cell-fate decisions and how do these cell-fate transitions occur? And we're, you know, both what I really, I think we're both driven to, and I know Prash succeeds in doing, is using a lot of different tools, but following the biology. So instead of being limited by a certain limited set of tools, Prash really goes in all these different directions where the biology takes him. And I find, I'm sure you'll see this in his talk, and I find it really beautiful. Everything from sort of cell biology to genomics to biophysics and biochemistry. And that's because he actually got his PhD at Hopkins in biochemistry, or in biophysics, sorry, from Sarah Woodson, and then moved to obviously the best model system, Drosophila, and did his postdoc with Ruth Lehman, and really started to develop this beautiful system in which he works, which is the germline in Drosophila. So he, after that, he started his lab at the University of Albany. I think it's called the University of Albany. There was wildly successful, won a Pew Fellow Scholarship and lots of great funding, wonderful papers. And building on that success, he's relocated back to New York City recently, and he's in the Department of, where is this? What? Oh, Department of Cell and Developmental and Regenerative Biology at Mount Sinai School of Medicine. And so I think this is going to be, I know there's a lot of really exciting stuff coming because he has like an intimidating number of bioarchive papers already, which means they're somewhere in the process. And I imagine we'll be hearing about some of them right now. So welcome.
Well, thank you for the kind introduction, Melissa. It's great to be here. I've followed literature coming out of here since I was a graduate student. There's a lot of RNA biology here and developmental biology since I was a postdoc. And since I started my own lab, a bunch of colleagues with impressive work. So I've never been to the Upper Midwest, at least on this side. So it's my first time in Wisconsin. So I'm super excited to be here. Thank you. Yeah. And I want to start by telling you that I'll be talking about the new transition in the germline, and it wouldn't have been possible without the people who actually did the work. Most of the work I'm going to talk about is done by two graduate students, current graduate students, Amy Sarkar and Noel Cook, as well as old previous graduate students, Alicia McCarthy and Patrick Blackwood, recently allowed to join various positions. And funding sources, past and present, for keeping the lights on for a while. So my lab works on germ cells. These are the cells that give rise to gametes, egg, and sperm. We work on germ cells because these germ cells, upon fertilization, give rise to all the somatic cells in your body, as well as new germline. Okay? And this new germline continues its march through generations, giving rise to a new organism in every generation. Therefore, the germline itself is pluripotent as well as immortal. So by studying the biology of germ cells, you can understand the molecular basis of pluripotency and, if you will, immortality. Okay? So among these germ cells or gametes, we specifically focus on the oocyte. Why is that? Well, many organisms require sperm for fertilization and launching the next generation. Some organisms have completely done away with it, and you can do this by a process called parthenogenesis. Okay, and also you can use somatic nuclear transfer by putting somatic nuclei into the enucleated oocyte, and you can start a new generation. Okay? So this shows that the oocyte alone is required and sufficient to launch the next generation. So there, again, by understanding the biology of oocyte, you can get a better handle on potentially developing regenerative therapies. Okay? So where do oocytes come from? As I told you, oocytes come from germ cells. These cells are among the first cells to form in a fertilized zygote. Once they form, what they do is they migrate through the embryo to reach the gonad. Once in the gonad, they have two options. They can directly undergo meiosis and make oocytes. This happens in women, or they can become germline stem cells that self-renew and divide and undergo meiosis to give rise to oocytes, which happens in female flies, which is the model system we use in our lab, and this is what I've introduced now. So each fly is going to jump to 3 slides further down, I think, hopefully. Right. So each fly has 2 ovaries. Each ovary is made up of individual units called ovarioles. These are assembly line of egg production. So the egg production starts at the anterior-most end in a structure called a germarium. The schematic is shown above and the pictograph is shown below. Okay. The germarium consists of broadly two tissues, the somatic tissue as well as the germ line. The somatic tissue provides structural support as well as signaling pathways that instructs the germ line. And in the germ line, the germarium starts with germ-line stem cells, which are present at the anterior-most end. Okay? These germ-line stem cells divide about once a day to give rise to a daughter, and both the germ-line stem cell and its daughter are marked by these round structures called spectrosomes. Okay? So once the daughter is made, it turns on a differentiation factor called bag of marbles, which is shown in green over here. Once it turns on the differentiation factor, it divides 4 times incompletely to give rise to 2, 4, 8, and 16 cell cysts. Okay, and the cysts are shown over here. Okay, and the cysts are marked by these extended structure called fusomes. It will show up. Fusomes that are shown over here. And once the 16 cell cysts form, meiosis starts getting initiated as differentiation starts from here. Okay, so meiosis, what is meiosis? You have the reductional cell division that initiates as well as recombination that initiates. So that starts in the cyst stages and eventually giving rise to a specified oocyte. Among the 16 cells, only 1 cell becomes the oocyte, and the rest 15 cells become nerve cells that provide mRNAs and proteins that help allow the oocytes to grow. Okay? So at least for the purpose of the talk, I'm just not going to use the clicker. I'm going to go behind the podium. At least for the purpose of the talk, it's important to remember that, okay, the stem cells and the stem cells in the immediate progeny are in the mitotic phase. This is where meiosis initiates in the meiotic phase. Okay, so once the oocyte is specified, what happens is the oocyte plus the nerve cells get encapsulated by somatic cells and forms the egg chamber. Okay. So once the egg chamber forms, the nerve cell starts initiating a lot of mRNAs and protein synthesis that gets loaded into the oocyte. This allows the oocyte to grow in size. Eventually, what happens is the nerve cells die back, leaving behind the mature oocyte. So this phase where mRNAs and proteins are made for the next generation is called the maternal contribution made of mRNAs and proteins. Okay. So my lab studies how to go from stem cells, enter meiosis, and eventually give rise to an egg. We have divided this process into 3 specific stages. One is the stemness program. This is stem cell renewal and differentiation. Next is the meiosis promoting program, which I'll talk about. And then finally, the maternal program, which gives rise to an egg that's capable of launching the next generation. We've published on all these aspects, how do germline stem cells divide and differentiate, how we enter meiosis and commit to it, as well as how do you regulate maternal contribution. Today, I'm going to talk about specifically program number 2, or the meiosis promoting program. So you may wonder what I call a meiosis promoting program. I call meiosis promoting programs the initiation of meiosis 1. So meiosis 1 is extraordinarily specific germ cell program. It involves recombination. So this recombination involves DNA double-strand breaks, crossing over, and such. This is, if you think about it, it's a crazy thing to happen to a cell, right? Not only that, and before I go tell you next crazy thing, the recombination is mediated by synaptonemal complex and various proteins that are associated with it. At least for the purpose of this talk, remember this protein called SUN, which I'll refer to over and over again, okay? So here is recombination. The other thing that happens during meiosis 1 is reductional cell division. This is you're having the number of chromosomes so that every generation you're left with the right amount of chromosomes, right? And in Drosophila, this is regulated by this protein called RB-FOX, something I will refer to again. So if you remember 2 proteins at this stage, these 2 would be helpful, okay? So the question we ask is, how is entry into meiosis 1 regulated? The reason why we ask this is entry into meiosis 1 is critical for all sexually reproducing organisms, and yet the understanding how this is initiated is poorly understood. To me, this is crazy, right? Like, this is the basis of all sexual reproduction. We still don't understand how this happens. And finally, exit. Exit we ask, I'll tell you in a few minutes after I tell you bit about how entry works. So we worked on the entry, and I will not talk about this that much. I'm going to summarize this, what we've done in entry in a few slides, and then go to the exit. So what we found in entry into meiosis 1 is 3 important things. One, that meiosis 1 promoting factors are present in the non-meiotic stages, the undifferentiated stages. This was a surprise to us and the reviewers, and this was actually verified by single-cell-seq, so that was good. So the mRNAs that promote meiosis 1 are also present in the germline stem cells and in the cyst stages, and eventually they start to go down. At least a cohort of them disappear. Okay, so this mRNA is present in these stages, and then, but these mRNAs are not translated. They are translated specifically in the meiotic stages, which is the cyst stages. Okay, so the first question we asked is what limits this meiosis 1 promoting factors mRNAs to the early stages of oogenesis. And that we found is licensed by expression of a chromatin reader that's only present in these specific stages. So again, I'm going to summarize a lot of data. So what we found is that a chromatin writer called SET2 is absolutely essential, and that deposits H3K36 methyl-3 marks. These marks are generally present on gene bodies. This is read by a factor that's only expressed in a temporal fraction called male-specific lethal 3 or MSL3. This reads these marks and recruits another complex called the ATAC complex. The ATAC complex deposits another mark called H4K16 acetylation. What this mark does is once it's deposited, it unpacks nucleosomes, allowing for transcription to happen. Okay, so once this mark is deposited, it does 2 important things. It does directly initiate transcription of various factors that initiate recombination, such as CONA, SON, and ART. SON, I remember at this point. And it also increases the level of a ribosomal protein called RPS19B. Okay. This ribosomal protein, what allows it to do is increase the level of ribosome biogenesis at that particular stage, which allows for the translation of a factor called RBFOX, which is, I told you, is the reductional cell division factor. Okay. So together, they coordinate entry into meiosis to allow for meiotic progression. So from here, we starting to address how transcription of meiosis 1 promoting factors is limited at these stages and is activated. We haven't by far answered all the questions as yet. So the next question we had is how is translation of these meiosis promoting factors only present at these stages? And this we found is regulated by ribosome levels. So in a completely unexpected twist, for me at least, what we found is that these meiosis 1 promoting factors have a lot of CAG repeats. So these actually code for polyQ proteins. We think this may help these polyQ proteins help assemble large synaptonemal complexes and so on and so forth. This is just a hypothesis at this point. But what happens is that these CAG repeats actually can stall ribosomes, okay? And we found that we need increased amount of ribosomes at this stage to successfully translate all these mRNAs. And this we published as a preprint. It's under review right now, so check it out. So now, so I've summarized to a certain extent how we're starting to discover how entry into meiosis 1 promoting program works in Drosophila. Now we're going to focus on the exit, which is what I'm going to talk about for the rest of my talk. So why exit? So first thing is meiosis 1 is an extraordinarily specialized program. You don't want things that cause DNA breaks, things that cause reductional cell division to be passed on to the next generation. So what was not known was that what happens to meiosis 1 products? Are they silenced or are they just simply diluted out? We didn't know, okay? So to address this question, I'm going to talk for the most of the talk about to the eyes of this one gene, if you will, which is the RPS19B ribosomal paralog that we talked about. So RPS19B is expressed in the mitotic stages as well as the early meiotic stages, and it's shut down once the oocyte is specified. The oocyte here is marked in red. You can see that as soon as the oocyte is specified, the RPS19B expression is shut down. Okay, this happens both at the protein level and the RNA level. If you zoom out and look at the whole ovariole, you can see RPS19B is expressed to the cyst stages and then shut off. Okay, the question we had was, what is shutting RPS19B that promotes meiosis 1 in these later stages of oogenesis? And one thing you can do in Drosophila is screens. You can do them fast, you can do them well. And so that's essentially what we did. And we found that RPS19B is transcriptionally silent at the stages where maternal contribution is being synthesized. And this we found, again, unexpectedly for us, It is mediated through a feedback loop between H3K9 heterochromatin and a nucleophore complex, which work together to silence these early genes that are present in the later stages of oogenesis. So how did we find this? Again, I told you we did a screen and we got ZDB1 to be one of the hits. So ZDB1 is a histone 3 lysine 9 trimethyl which deposits this mark, allowing for heterochromatin to form, causing gene silencing. If you lose ZDB1, you lose these marks and you get expression of gene expression, okay? So one crazy thing about ZDB1, which had been known before, but it's not fully appreciated why it does it, was that it's cytoplasmic prior to oocyte specification and becomes nuclear after oocyte specification. So there's kind of translocation that happens at this stage when meiosis 1 is initiated, okay? And this translocation is mediated by a conserved protein called Windei. If you're a German speaker, I really apologize. There's a lot of German names which I'm going to butcher, okay? So Windei, which allows for translocation of ZDB1 from the cytoplasm into the nucleus. So in our screen, we found that ZDB1 and Windei are both required for silencing RPS19B. Here you can, so here you can see RPS19B expression in the germanium and is shut down in the later stages. But if you lose ZDB1 in the germline, now RPS19B is blazing in all the later stages, as well as losing Windei, which blazes again all in the later stages. This shows that ZDB1 and Windei, which means the nuclear translocation of ZDB1, is required to silence RPS19B in the later stages of oogenesis. So next question is this RPS19B specific, or other early oogenesis genes are also regulated the same way, okay? To address this, we carried out RNA-seq by specifically depleting ZDB1 and Windei in the germline, and we found about 800 genes to be upregulated, okay? And among these genes that were upregulated are genes that mediate cell differentiation, suggesting that things that promote differentiation are not being shut down, okay? So a prediction is that if this is true, then you should see these genes normally being expressed in the early stages when things are differentiating, and they should go down. And in ZDB1, these differentiation genes should not be shut down, okay? So to do that, what we did was we stopped development. You can do this in Drosophila, stopped germline development at various key hallmarks of germline program differentiation, and we enriched for those particular stages. Okay, so we have GSCs, we have GSC daughters, we have cysts, we have young wild type, which are the first few egg chambers, adults, which allows for later egg chambers, and so on and so forth. And then we plotted the genes that are upregulated in ZDB1 and Windei together as a function of development. So as you can see that there is an increase from the germline stem cell stage to the cyst stages. And then once you hit the cyst stages, it goes down. Okay. Oops. It goes down, okay? But if you lose ZDB1 or Windei, this does not happen, showing that cell differentiation genes that are present in the differentiating stages are being shut down in the wild type, but it's not being shut down when you lose ZDB1 or Windei, okay? So from here, we concluded that nuclear ZDB1 is required to silence germ cell and early oogenesis genes. You may be asking why I keep saying germ cell. So one thing I didn't tell you about is that a lot of genes that we saw that were silenced didn't actually have a role in cell differentiation, but they were merely expressed in the early stages of germ cell development, which apparently have no role in female germline, but they were being shut down as the oocyte are being specified. If you are a Drosophila aficionado, it includes genes such as blank. So we think that these are like germ cell genes, that primordial germ cell genes that have a hard stop at that particular time point. So, okay, we know now that ZDB1 is required to silence these genes. The question is any of this direct, right? So we predicted that if ZDB1 directly silences meiosis 1 promoting genes, then it should deposit some kind of H3K9 mark in and around this meiosis promoting gene, allowing for silencing of this gene to happen, okay? So to test our prediction, we then carried out cut and run of H3K9 methyl 3 marks, and we saw its distribution over the entire genome. Again, for the purpose of this talk, I'm showing you these two loci because it's just easier to follow than the whole genome. Right, so here's an RPS19B locus. You can see in wild type there is beautiful heterochromatic marks present, allowing, and I have to say that these were enriched for the later stages of oogenesis. So we are enriching where it should be silenced, and at those stages we see an accumulation of these heterochromatic marks present in RPS19B, and which is then lost when ZDB1 is lost. And this also happens in the some locus, which is allows for recombination to happen. We have presence of H3K9 methyl 3 marks around the promoter, which is reduced in ZBB1. Okay. From here, we concluded that H3K9 methyl 3 heterochromatin silences germ cell and early oogenesis genes at the onset of oocyte specification. So this could have stopped right here. I said, we figured this out. This is great. But we found something else that was really puzzling to us. And this, we opened a Pandora's box in a good way, right? So here, as I told you, things that were upregulated included cell differentiation. But we looked at RNA-seq of downregulated genes in WND and ZBB1. We found nucleoplasmic transport to be downregulated. All the other kind of downregulated terms made sense, but nucleoplasmic did not make sense at all. Like, I was like, what is going on here, right? So as you know, nucleoplasmic transport is mediated by the nucleopore complex. And the nucleopore complex allows for transport of cargoes in and out of the nucleus. It's made up of several proteins called nucleoporins. About 38 nucleoporins are present that comprise of this complex. We found that 18 out of 30 nucleopore complex members were downregulated when there is no heterochromatin. Okay, so this is important direction to remember. When there is if there is heterochromatin, there is nucleopore expression. If there's no heterochromatin, there's a decrease in 18 members. Okay, this intrigued us for many reasons, including the fact that in yeast, recently it's been shown that nucleoporins are part of the heterochromatin proteome, as well as nucleopore complexes allow for organization of heterochromatin at the nuclear periphery. Okay, so this forces us to ask two questions. The first question was, is it possible that when you lose heterochromatin, you're losing nucleopore complex formation? If you think about it, it's a little bit wild, right? And the second question is, does nucleopore complex regulate heterochromatin formation in Drosophila like it does in yeast? So to answer the first question, we stained for nucleopore complex using a marker for NPC, which recognizes multiple proteins of the nucleopore complex. It's just not one. Okay. When you look at wild type, you see beautiful nucleopore complexes that are present in late stage oogenesis. You can see them here. Okay. And when you lose ZBB1 in the germline, this is losing heterochromatin in the germline alone, you see complete loss of nucleopore complex. You form these punctae. We don't know what these punctae are, and neither do we know how these cells live without nucleopore complex. If you ask me, this is crazy, but that's a whole nother different Pandora's box to be open. But nevertheless, we concluded that heterochromatin is actually required to form nucleopore complex in the nerve cells of female germline. The next question, of course, was what happens to heterochromatin when you lose nucleopore complex? So for that, we stained for H3K9 methyl 3 in wild type, and upon depletion of a specific nucleoporin, which destroys nucleopore complex formation. Okay, so in wild type nerve cells, what we see is this heterochromatin that's present at the nuclear periphery that's attached over here, and you can see that dense region. While if you lose nucleopore complex, there are two things that happen. First, this heterochromatin comes off the nuclear periphery. It's often present in the middle of the cell. And eventually this thing dissolves and it goes away. So which is shown in the next slide here. So if you look at nerve cell nuclei over there, that has no heterochromatin present. And this can be quantitated and can be put as a function of development. You see that in egg chambers that are present, eventually you lose heterochromatin. So from here, we concluded that you need heterochromatin to form nucleopore complex. And this nucleopore complex then is required to maintain heterochromatin. Okay? So next question we asked was, okay, this is a feedback loop. Then if this is true, then in absence of nucleopore, we must also lose expression of these germ cell and early oogenesis genes, right? So again, to test that, we depleted one specific member of nucleopore complex, which is not 154. I should add that we've done this with multiple nucleoporins and we see the same effect. So again, in control, we see RPS19B expression present only in the early stages of oogenesis and then shut down. When you lose nucleopore formation by knocking down NUC154, you see expanded RPS19B expression. We can do this with RNA-seq and show that it shares about 97% of the targets with heterochromatin, with ZBB1 as well as WND, which allows the heterochromatin to form in a regulated fashion, showing that a large fraction of targets overlap with the nucleopore complex. Right. So the last question we had was, if this is true, then NPC should directly affect heterochromatin formation on these specific loci. Again, I'm going to show you those two loci, which I've been showing you. Is now look at RPS19B heterochromatin, but now when we knock down NUP154. When we lose NUP154, again, we lose heterochromatin on the RPS19B locus, and it's reduced at the sun locus. You can do this with overlapping peaks, and we see about 90% of the targets that have peaks are enriched for H3K9 peaks are being lost in NUP154, showing that the targets do overlap. Okay, so from this part of the talk, what I want you to take home is that there is regulated heterochromatin formation during oogenesis. This regulated heterochromatin formation allows for nucleopore complexes to form at that particular stage. This nucleopore complex then allows for heterochromatin maintenance. Together, they silence meiosis I promoting genes, which allow for this transition from germline stem cell to an oocyte. Okay. So, of course, this is great. This model is nice, right? But then we now say that when you lose this whole feedback loop, you lose this regulation. So what happens? Why do we need to silence the specific genes at that particular time point? So for that, what we then did was we looked, so this is the meiosis I promoting program. And I told you after meiosis I is the maternal program, okay? So we asked what happens to the maternal program if the meiosis I program is not shut down, okay? For that, we used a specific antibody against oocyte called egalitarian, okay? The egalitarian is an RNA binding protein that binds maternal mRNAs and transports it to the oocyte. If you look at egalitarian staining, it's beautifully localized in developing oocyte, okay? But if you lose ZBB1 or nucleopore complex, there are two things that happen. One, you have egg chambers present, but they do not grow in size like they do in wild type. And they initially specify an oocyte, but the oocyte specification is not maintained, okay? So this suggests that silencing of meiosis I promoting genes is absolutely essential for a proper maternal program to be executed, right? So with this, I would like to summarize this part of the talk where we have now shown you that there's germline stem cell fade and meiosis I genes are silenced as the oocyte is being specified, mediated by H3K9 methyl 3. And this silencing is absolutely essential for the oocyte to be formed. Just need to change the battery. Sorry, I don't know why it's dying, but there's another one. Yeah, I have a battery. Sorry. This is useful when undergraduate teaching is you always carry extra batteries. Yep. So this essential is allowing for maternal contribution to be made. And the second conclusion is what kind of unexpected for us was that heterochromatin and nucleopore complex co-regulate each other. You need proper heterochromatin formation to form nucleopore complex and proper nucleopore complex to allow for heterochromatin to be maintained. Okay? And this led us to potentially extrapolating how this may work in other cell systems. So this led to a curious model, right? So every cell has a different amount of heterochromatin in there. And so heterochromatin needs docking sites to be maintained. So now we have a system where heterochromatin dictates how many nucleopore complexes there will be there. And these nucleopore complexes then we can maintain heterochromatin at the very periphery. So you have a model, you can scale up and down the heterochromatin docking sites and heterochromatin different cell types based on this feed forward or this mechanism, all right? So this summarizes this part of the talk. And now I'm going to switch and ask a question. Now we know that meiosis I genes are being silenced. How in the world are they being specifically targeted for silencing? I have to confess and tell you upfront, this is an incomplete story, but I think we have the answer. All right. So we already know these genes are being silenced. The question is, how does heterochromatin get targeted in general? Do we know anything about it? Okay. In humans, heterochromatin deposited by ZBB1 is targeted to specific loci mediated by zinc finger proteins called CRAB-ZFP proteins. These are fast evolving genes that are present in the human genome. There are lots of them, and they have unique identities and functions. Okay? Then you can say, let's just look at crab ZFP in Drosophila. This should be the answer. However, there's a problem. Drosophila does not have crab ZFP proteins. All right. So this led us to how in the world then is ZDB1 targeting these genes for silencing? Luckily for us, work from Harmit Malik and others have shown that ZAD-ZFN proteins, another family of zinc finger proteins, act I can't say this word, to crab ZFN proteins in flies, okay? The only problem is that there are 91 proteins. Tell a graduate student, you have to screen 91 proteins at the end of a PhD if you're gonna shoot me, right? But luckily for us, somebody had already done the work, right? And so Malik Lab, Harvard Malik and Helen Saul's lab have done a screen of ZAD-ZFN proteins where they look to see what their general effects are on germline. So we already know that if we don't silence meiosis 1 genes, the chambers do not grow, and they express RPS19B. So what then we did was just look at the phenotypes and said, okay, we'll just pick ones that have this repeated egg chamber phenotype where egg chambers don't grow, and then screen those alone for RPS19B. And that's exactly what we did, and we found that ZAD-ZFN protein knickknack is required for the silencing. So I'm just going to show you one picture. So again, you have control over here, and you have silencing of RPS19B. If you lose knickknack ZAD-ZFN protein in the germline, you get this RPS19B expression. We can verify and validate this with other candidate genes. However, unfortunately, I was hoping to have the RNA sequence and hadn't run back by the time I came here. They're still in the process of sequencing it. So I think we have the answer, but unfortunately, I can't extend it to the whole genome as yet. So from here, what we are finding is that ZDB1, in a very analogous manner to what we find in mouse, are targeted to specific loci in the Drosophila genome by knickknack to silence early oogenesis genes. Okay. So luckily for us, there are 2 papers that came out this year, one from Saul's lab and one from Julius Bernatchez lab. From Saul's lab, they find that a gene called identity crisis silences male genes in the female germline. And Julius Bernatchez shows that another ZAD-ZFN protein called Kipfer, sorry if I'm butchering another German name, silences transposon and piranide clusters. This shows this family of 91 ZAD-ZFN proteins may be doing some unique roles in the germline during development. Okay. Do you have any questions before we move on? What's that?
I have a question, yeah. But I could say you could say the end at the end. Okay. Because I was just wondering when you see this connection between no heterochromatin and then the decrease of nuts, and you are potentially affecting the nucleoside plastic transport of a ton of different things. So because and then the growth of the follicle is arrested and you see loss of pairing, but that could be even if you are not getting proper export of mRNAs for all kinds of things, and transporters and
the phenotype could be because of anything, right? But what strangely we find that ZDB1 still translocates when you don't have nuclear pores. I don't know how this happens. With our nuclear pores, they're completely fine from stage 1 egg chambers to stage 7. How these cells function with our nuclear pores, I have no idea. How they export. The GFP comes out of the nucleus. So if you look at the pictures, I can show it to you again at the end of the talk. RPS19B comes out and goes back in the nucleus. So they're able to come out, be translated, and the GFP goes back in the nucleus. So at least for select candidates that we've tested, nuclear plasmid transport is working. Maybe it's very low levels of nuclear pore complex that we do not see that's doing this function, but I still don't understand how these cells are able to survive even with those low levels of nuclear pore complex. So this I don't understand the biology at all. There's something strange going on. We have to get to the bottom of this. So, but it's not simple as nuclear transport is affected because we do see things going in and out. How this happens, I have no idea. All right. So next, I'm going to switch. So from mechanism of how, first we talked about silencing genes, meiosis 1 genes in later stages. Second, we talked about how these genes are being targeted specifically for silencing. third question we ask is, how do you get this transition? Why at this particular stage do you get this transition, right? What's happening over here that allows for germ cell enriched genes to be silenced, allowing for the oocyte program to be started, okay? So here is the green, you can see germ cell enriched with RPS19B, silence, and the oocyte enriched. So why in the system? Okay. And then again, if you know the answer already, I'll tell you that we did a screen. And again, the answer we expected was not the answer we were thinking it would be. We found that increased ribosome levels at that particular stage allows for this transition to be mediated. And what the increased ribosome levels do at that particular stage is allow for translation of particular components of the nucleophore complex, allowing for this transition to happen. You may be asking me what in the world is going on? This is the exact question I had. And so we'll go through slide by slide, okay? So when we carried out a screen of what silences RPS19B GFP expression, we got a whole bunch of genes that are involved in ribosome biogenesis, as well as and TARC1 pathway, okay? So for example, we get ribosome protein ZFRP8 and bistin, which are involved in ribosomal 40S, 60S assembly to be involved. We also got members of the TARC1 pathway, which such as Raptor and Mio to be involved. Okay, so the idea that these proteins were involved in mediating this transition came from looking at activity of TARC1. Okay, so TARC1 is a kinase. It phosphorylates various different proteins, including ribosomal protein RPS6. Okay. There are several targets, and it allows for ribosome homeostasis to be maintained. If you look at phosphorylated RPS6, you see increased levels in the cis stages where the oocyte is being specified. Okay, so this shows that TARC1 is really active over here, and if you and this happens exactly during the transition. Okay, so what happens if you lose TARC1 and ribosome biogenesis? So we found that again, like in control RPS19B silence in the later stages. If you lose Mio, which is a negative regulator of a negative regulator of TARC1. So bear with me, this is a complicated process. TARC1 provides phosphorylation activity. It's repressed by Gator1 complex. Gator1 complex is repressed by Gator2 complex. So if you remove complex of the Gator2, Gator1 will repress, right? So if you remove Gator2, then you have no TARC1 activity. And when you do that, you get expression of RPS19B. If you now remove members of the TARC1 complex itself, such as Raptor, you get RPS19 expression in the later stages of oocyte. This shows that you need increased ribosome homeostasis at that particular time point mediated by TARC for silencing to happen. This is not just mediated by TARC. We can knock down various aspects of ribosome biogenesis. I'm showing you two here. Oops. Bistin, again, you get these expansion of ribosome biogenesis. It's possessed, sorry. Okay. And then if you lose ZFRP8, you saw it. It doesn't silence RPS19B expression. So from here, we can conclude that ribosome biogenesis and or translation is required for silencing meiosis 1 or early oogenesis genes after the oocyte is specified. The question you may ask, how in the world is this happening, right? So to do that, what we did was we carried out a polysome-seq analysis. So as you know, polysomes are actively transcribed mRNAs, and you can actually purify polysomes from cells. Okay? You can do a density gradient from light to heavy, and you can specifically isolate polysomes. The idea here is that what happens if you have wild type is that you would have regular mRNAs being translated, and if, under ribosome limiting conditions, there should be some mRNAs that are not being translated properly. Okay, so you can purify these polysomes, and you can say, okay, what are the mRNAs that are not being translated properly? So we did this in wild type and ZFRP8 conditions. You can see already in ZFRP8 conditions, ribosome biogenesis does not happen properly. Then what we did was we take the mRNAs present in the polysome, and then we sequence the total input in those very same cells. So you can compare what is the total amount of mRNAs present in a cell, and what fraction of that is associated with actively transcribing ribosomes. Okay? And you can plot this, and you can calculate translation efficiency of that particular stage, at that particular stage of those mRNAs. And we did that, and the answer we got, again, was a big surprise to us. When we calculated translation efficiency, we found that several NUPs were downregulated at the level of mRNA, or at the level of mRNA and protein, or at the level of translation alone. So basically, there's complete loss of homeostasis of nucleophore at that particular stage when ribosomes are not being there. Okay, so this led to a strange hypothesis. Is it possible that if you don't have enough ribosomes, you don't make enough nucleophore complex? This, again, if you think about it, it's kind of crazy idea, right? So that's exactly what we tested. We looked at control, where you have beautiful nucleophores forming in the later stages of oogenesis. If you knock down genes that are required for proper ribosome homeostasis, such as MEOs, DFRP8, bistin, or whatever, you lose nucleophore formation, nucleophore complex formation. This is kind of crazy, right? So from here, we concluded that, okay, RPS19V transcription is silenced, and we know that there is this feedback loop that's present, and ribosome biogenesis allows for nucleophores to be translated properly, which then presumably allows for heterochromatin to form, then allowing for silencing of early oogenesis genes. If our hypothesis is true, then we should, in ribosome biogenesis mutant, you should now lose not only nucleophore complex, but also proper heterochromatin formation, right? So that's exactly what we tested. We asked, what happens to H3K9-methyl-3 marks across the genome when you lose proper amounts of ribosomes that are in a cell? Okay. Again, we did cut and run, and then I'm showing you two loci, RPS19V loci right over here. You can see enrichment of H3K9-methyl-3 marks. This is dramatically reduced in a mutant that has nothing to do with chromatin, but rather to do with ribosome biogenesis. Again, if you can look at ZFRP8 mutants, you have beautiful enrichment of RPS19, heterochromatin RPS19 locus over here, and you lose that when you, again, lose another component of ribosome biogenesis. These have nothing to do with transcription, chromatin whatsoever, okay? So from here, we conclude that at this transition, increased levels of ribosome biogenesis allows for translation of the nucleophore complex machinery. This nucleophore complex machinery allows for heterochromatin to be maintained, which is being made at that particular time point. If you remember, it has that DB1 localizes to the nucleus from the cytoplasm. And this allows, and then you have a transcription factor that's present that allows for silencing of these early oogenesis genes that are present. Okay. So this was exciting to us for many reasons. First reason was that TARC1 is known to regulate entry into meiosis from yeast to humans, right? So now we are saying that TARC1 not only regulates meiotic entry, but also makes the machinery that silences the meiotic machinery after meiosis is done, or like after meiosis is initiated, right? Not done. So that's one idea. Second idea, what I'm really, really excited about is that now we are saying that ribosome levels, changes in translation within a cell, can be encoded at the level of epigenetic changes at the level of transcription, right? So we are changing ribosome levels, which then changes nucleophorins and NPC levels, which then causes changes in epigenetic state of a particular cell, right? So this is a cool idea because neurons often change levels of translation based on long-term memory and so on and so forth. And potentially could get encoded at the level of transcription, all right? So this begins to conclude my talk. We talked about entry, we talked about exit. And I told you exit so far, all transcriptional exit, right? How do we silence transcription of these meiosis-1 promoting factors in the later stages of oogenesis? One question we had was that what happens to the perduring RNAs that actually code for these factors? Such as, again, you have the recombination machinery that's already made, right? The mRNAs are still there. What happens to these mRNAs? In a published work, I'm not going to talk to you about, I'm not going to show you more data, is that we've already shown that regulated RNA degradation that happens at that particular time point clears the early mitotic and meiotic program mRNAs when the oocyte is getting specified. It's as if when the oocyte is being made, you want to get rid of everything old and then start everything new again, right? And so I think for today's talk, what I've told you is that in germ cell, you have germ cell mRNAs, you make it by a chromatin axis, which is the reader writer I told you about. And then you translate these things with increased ribosome biogenesis because CAG repeats require increased number of ribosomes. And then what happens is that this needs to be silenced, okay? This silence is mediated by heterochromatin, nucleophore factor, and a transcription factors, one of which seems to be a ZNF-ZAD protein. And then once you silence transcription, you remove all these mRNAs mediated by mRNA surveillance pathway called the no-go decay pathway, okay? All this is absolutely essential to launch the next generation, to allow for the maternal contribution to be made. Because if you don't silence this, if you don't remove the mRNAs, what happens is the next generation, stuff for the next generation cannot be made properly and you end up dying, okay? So I want to show you this and then zoom out. What we are finding is that we've discovered a new transition that we term germ cell to maternal transition. I'm getting a lot of pushback for the name. You can come up with alternate names and be happy to take ideas. So what we think is happening is that the oocyte fate is bookended by two transitions. One transition you probably heard of extensively because Melissa works on this is maternal to zygotic transition, right? Here, maternal mRNAs are degraded, allowing for zygotic transcription to happen, which Harrison lab works on. And what we are finding on the other side, a flip side of the maternal mRNA contribution synthesis, you have all these germ cell mRNAs, meiosis mRNAs, and stem cell RNAs that are transcriptionally silenced. mRNAs have gotten rid of to allow for proper maternal mRNAs to be made. So we think that germ cell to maternal transition, maternal to zygotic transition, bookend initiation of a totipotent fate and ending of a totipotent fate, if you will. Okay. So while I've told you everything that is that happens, that involves, right? That involves we will get to questions eventually. Oh no, next previous slide is an important slide, but okay, so at that particular transition, oh, there it is. So we find transcriptional reprogramming that happens, right, which I told you about, then mRNA selection that happens. What I didn't tell you is that there's a whole bunch of mRNAs that are separate to earlier stages of germ cells. These are present only in the germ cells, germline stem cells, and the meiotic stages. These ribosomes are destroyed. We have an idea, we have a hit from a screen of what that may be. And then also, strangely, we're finding that old nucleophore complexes that are present in the early stages also destroyed, allowing for new nucleophore complexes to be made. We talked about synthesis of new nucleophore complexes. So I think what we are finding is that all the old stuff gets destroyed over here. And Hurd and Lehman groups have already shown that there is a mitochondrial selection that goes on at that particular stage that allows for non-defective mitochondria to be selected. So what we really think this transition is about is cellular rejuvenation. So if you think about it, it doesn't matter how old your parents are, you always start the brand new life at age time point 0, right? So at some point, you got to get rid of something old and start something new. And we think this at maternal germ cell to maternal transition is when this happens. Hard reset happens. Everything old is gotten rid of and everything new begins so that you can start new generation of fresh with undamaged goods. So you don't carry any baggage from the previous generation. So I will stop here and questions. And so one advertisement before, if any of you know anybody who's looking that thinks this may be interesting, please do contact me. Yeah.
So you were talking about the amount or bulk of ribosomes driving translation. Can you exclude possibility that when you're making more ribosomes, you're making specialized ribosomes that are different than the ones before that?
You know, we've searched hard for like specialized ribosomes. When I got RPS19B, I was really excited. We can rescue that with just putting human RPS19, rescue that with just RPS19A. We've done this with autoparalogs. So we don't have any evidence for specialized ribosomes, unfortunately. You know, that's very popular, you know, but we can, at least genetics-wise, what genetics tells me is that you can rescue all these paralogs with autoparalogs. So it really, why are these paralogs existing, you may ask? So just, this is a strange phenomenon of observing that I think it also happens in C. elegans. There are ribosomal paralogs on the X chromosome and the autosomes. So what the paralogs we see expressed are on the autosomes when you have these autosomal paralogs present on the X chromosome. For example, RPS19A is on the X chromosome, B is on the autosome. So we think this may be a compensation mechanism due to like meiotic X chromosome silencing. That's just like a leftover thing that's happening that may be causing these things. I don't think this has anything to do with ribosome analog.
So you just think the bulk of ribosomes is now increasing scanning and increasing, the loading onto the particular mRNA?
Right. So, so how do nucleophore? So I don't know about generalized view of other mRNAs, right? How do nucleophore get increasingly translated due to increased ribosome levels? So what we are finding is NUP44A has a top motif, which we recently identified that top motifs are present in Drosophila. And top motifs are actually sensitive to ribosome levels. So it so happens that nucleophore complexes are coordinately regulated in terms of translation and transcription. And so we think that NUP44A has a top and allows for it to be sensitive to translation of ribosome levels. And so if you lose translation of NUP44A, you destabilize other RNAs because they're all dependent on one another for their translation. Am I making sense? Yeah.
My question. Could it be that rather than the ribosomes, what's changing is something like 550 properties to the cytoplasm and cell crowding that is affecting the formation of things like that?
I don't know, I mean, yeah. Right, I mean, so I agree that I believe in genetics. I'm a firm believer, genetics is telling us that, I mean, you mess up ribosomes, you mess up a lot of different things. It could, and I know there's, there's evidence that plasticity changes at that point. Any of that could be a possibility, but at this point, genetics tells us there's one set of protein, one set of things that are messing up things up in the ribosomes. Yes.
Between the 16 cells, how they decide which one will become the oocyte?
Right. This is a long, big question that's still left. I don't know the answer. I think there was a recent paper from St. Johnson group showing that one critical protein that's blanking on the name right now, sorry, but allows for this oocyte reset, right? So the idea is that oocyte, you get 16 cells. 2 cells have the maximum number of connections between other cells. So that those two become the pro-oocytes. Between those two, there's a competition for this protein, and then one cell wins out, allowing for that cell to become the oocyte. That's the model right now. But it's not fully clear how we pick a oocyte. The question is, does the oocyte have the cell that actually has the most refreshed material become the oocyte? I don't know. That's one of the ideas that they're throwing out.
Right. I'm going to be a pain.
Oh, no.
Yeah, because I mean, your data show those connections, right? But I just think that there's so much more going on, because if you talk about, even if you see that some proteins are going through the nucleocytoplasmic transport normally, there is like a lot of different types of different sizes, different types of nucleocytoplasmic transport. So it's unlikely that it's going to be like one thing. Right? And then the same thing with if you decrease levels of ribosomes, yeah, you affect production of lots of different things, including nuclear proteins. But yes, just how do you? Because I know that the kinds of experiments that we think of as genetics is like, how do you test that in states, or how do you so how do you show that that's really there's a cause-consequence relationship that is really difficult. So I'm not even waiting, but I'm just thinking, how are you going to continue this?
Right. So first thing would be the genetics idea, right? Knock down one particular. So if I say ribosomes affect NUP44A, right? Hundreds of thousands of things can go wrong, but we reduce to NUP44A and we get loss of protease expression, right? But when I say NUP44A expresses hundreds of different things, right? We can do this with every single protein we see, knock it down specifically, not just ribosomes. Knock it down, we see the same thing, right? So I agree that, of course, cells are not well if you reduce ribosomes or reduce nuclear pores. But I think the effect that we get this RPS19B is clear in terms of what is happening. And there could be thousands of other things going wrong. But we think this derepression is because of that particular.
Yeah. And also just like another layer of complexity, right? Because it's not just a change in levels of expression of the RNA or a change in how much protein is made, but they're just, and related to Francisco's questions, there's like so many other things that change neurogenesis that might not have anything to do with the levels of anything, right? It could be just substantive localization, all kinds of different types of regulation, maybe in this case. I have my own biases.
Right. So I think the question I think you're getting at, and I agree with you. How do you get specific effects on something that's changing things broadly, right? And so
it's just, I don't think it will be hard to have like a simple model like that explains this transition. Like it just seems like a much more fluid system.
I agree, but we can knock down single genes and get the same effect.
Well, to follow up on that, I mean, maybe I missed it. Did you look at other opportune markers and see if they changed?
So that's great, right? So we look at H3K27. So if you lose nuclear pore complex, it's not as if all the chromatin marks are going down, right? K27 actually increased. And K4. So we actually know, this is a great question. We know how loss of heterochromatin actually affects nuclear pore complex, which I didn't tell you because what happens is that you have these domains of K27. And when you lose K9 heterochromatin, K27 heterochromatin goes up and spreads. And the nuclear pores, the 18 nuclear pores that seem to be present are present within domains of these K27. So this increase causes a decrease in nuclear pore complex, resulting in loss. So we know mechanisms and identities. So we know this is not generalized broad-based view of things going wrong, but rather specific mechanistic things that we can test. So of course, now we don't have this experiment, which would be useful is put back NUP44A and rescue it, which is in progress, and which we're doing with EZ and PRC2 for SETD1. I think that would be the same. But yes.
So then a follow-up to that, I mean, so do you think this kind of sensitivity to, I mean, a very generalized kind of insult, this generalizes to other kinds of cells that you see the same kind of feedback loop, or
We have done that.
I was discussing for years.
No. I mean, so it would be great to collaborate with other people. Like I know, I mean, when I'm trying to like already we're going nuclear pore chromatin RNA, so I'm trying to keep it somewhat focused in my lab, and so like let's stick to the female germline. But that being said, it would be great to look at this other cell types. We haven't done this experiments, yeah.
And we love this but I was just another off the cuff thing is just when you see this perpetuation of the early expression, what happens to the fusome? Because the fusome
it goes away now. Yeah.
Follow-up question of that comment is like really the germline Markov condition to apply to like other kind of stem cells, differentiated cell, like similar.
Right. I don't know. I mean, what hope is that we're starting with somebody in mouse genesis, right? The hope is that these things are conserved, and we have to look. We're still like understanding the biology of the female Drosophila germline, and so the next step is to actually look in other cell types, other germlines, and see. I mean, we're 100% convinced that there is a meiosis 1 level mRNAs that are down. There's no question. And whether this is conserved in other organisms, I don't know. This is something we are against, and we need to look at see if it actually happens. And we need as many collaborators as we get. Yeah. There's a question about.
So I'm wondering how the regulation of the meiosis promoting RNAs interfaces with the nanos.
Nanos, I don't know the answer. I should, because I feel like worked on this.
I mean, it's not something you should know. I'm curious, and I think other people will also be curious. So I want you to be prepared for that.
Right. So what we haven't done, and I think it's a great question, to look to see how many of these have like, FBF binding things in the three primary GI.
And my bet is that they all have. I bet that it's really important.
I think you're absolutely right. And we haven't done that.
But you're talking, at least in your seminar, it sounds like you really understand everything in cell transcription, and yet you're just leaving out.
No, I was.
I think it's really important, and a lot of other people think it's important.
I was a translation biology person. So, but often, people. The reason why I want to say this case is that often people say transcription is not instructive in the journal, right?
Yeah,
right.
I don't buy either.
Right, and so now we're showing an example where transcription is actually instructive in the journal, right? And so from
have a little clause and not disregarding.
Yeah, I would say, it's allowed from Kumolio and I completely agree. But so here's the thing, right? You have the translation machinery, right? And then you have the licensed mRNAs for their translation. So of course, we're coming at it just from the translation machinery point of view. We haven't quite asked it what mRNAs have licensed to be translated by Kumolio and Bruno and other things at that particular time point, right? Yeah, I mean that needs to come together at some point. No, quite well taken. No, I mean, often I know you know this, right? But when people say transcription germline is not instructive, there's just transcript pervasive. That's not the case at all, right? And we are finding that a significant portion of the genome is transcription regulated, and significant portion of the genome is regulated at the level of translation, right? And so at this point now we have to figure out how they interface with one another. So
that's why that's the interface word is.
That's great. I'll use that. Thank you very much. If somebody says that Judith Kimball told me that, yeah. Any more questions?
So have you looked at nuclear valence and how it's affected?
It doesn't. And so it's one of the reviewer questions as well, right? At least we haven't done EM by just lamin C staining. It doesn't seem to change, right? And so I don't know if there are minute differences, but gross level.
When you're going from block reports to very few, you might expect an effect on it.
Right. I mean, also, if anybody has any ideas, please shoot me an email. How cells function with an individual component.
I think it's really not.
No, but even
or it's very low.
Very low. But if you then why have so many nuclear pore complexes in the first place, right? I guess for chromatin, then most of the nuclear pores are required for chromatin regulation. Yeah. That's what's been bothering me. Is just like then you don't need all these nuclear pores to transport stuff in and out of the cell. You just need it to like maintain a chromatin. Right. Yeah, exactly. And so, but these, I don't think they found annulate laminate later on that actually are deposited maternally. So I don't know what happens to the nurse cell.
Oh yeah, they just get trapped.
Right. Yeah. Right. Exactly. And so the nurse cells get trashed along with the nuclear pores, I think. But you're right that the maternal store is being made at that particular time point. Any more questions?
Just a comment about that. All that thing, why makes so many, but you're thinking of the steady state condition. You never know when you might need a different setup.
No, it is fair. But I don't know, I think, I don't know, we look at, we stay for nuclear pores and cell types, we see a lot of them, and here we, at least visually, you don't see a lot of that. They seem fine, and then they fall apart later on. So.
Maybe they're storage.
Right. Yeah, exactly right. And that's I don't know what happens. And so what we're really doing, and I think I'm super excited by these experiments, we are actually tagging these nuclear proteins with timer proteins like old and new. And so we can actually now look at old ribosomes, new ribosomes, old nuclear pores, new nuclear pores. And we're doing tagging on a lot of different cellular components with these timer proteins, hoping to see what happens to them as functional line. And it's really like there is a rejuvenation of cell cellular component that is really good. Fingers crossed, we'll see if it works or not. Thank you, everybody. Oh, this. Thank you. Yeah.
Trying to give you a hard time.
No, but it's always like,
Having a lot could be all about selecting.
Right. Yeah.
Right. The
Yeah,
selection.
and I mean, there's like, often it's something to think about, right? Specific effects of things that change a lot of things.
Yeah.
Right? And so how does that happen? How do you get
Yeah.
that, right? And
but this idea of having way more nucleophores than you need, like apparently,
right,
could be just to select good ones later.
Exactly. And so that's what we're hoping to see, right?
Yeah.
See.
Anyways, great job.
Yeah.
See you in the next
Yeah, I know.
Thank you for the slides.
Yeah, no problem.
I will email you later to ask you the full genome.
Yep. And yeah, let me know if there's any issues.